Hello and welcome to today's lesson. In previous lessons we've looked at genetic definitions, we've looked at genetic crosses, and today we're going to look at genetic engineering. So if you could write in your book the title, as you would do for any other lesson, genetic engineering. By the end of this lesson you should be able to define what a genetically modified organism is. You should be able to explain the process of genetic engineering to produce genetically modified organisms, and you should be able to evaluate how genetic engineering could potentially be used to cure people with inherited disorders. So your title is genetic engineering. Let's make a start. So the first key word I want to get out of the way is the term genetic engineering. Genetic engineering. Now, genetic engineering is a term used to describe the transfer of specific genes from one organism to another. So if we are transferring genes from one organism to another, we are carrying out genetic engineering. Any organism that goes through genetic engineering is known as a genetically modified organism. So we can say any, that's not how you spell any, is it? Any organism that undergoes genetic engineering is known as a genetically modified organism. Is known as a genetically modified organism. And that brings us to our next key word, genetically modified organism. Genetically modified organism, or GMO. So these are frequently abbreviated as GMOs. Okay? A genetically modified organism is an organism that has had new genes transferred into it. Transferred into it by genetic engineering. By genetic engineering. So genetic engineering is when we transfer one gene from one organism into another. Any organism that undergoes genetic engineering becomes what's known as a genetically modified organism, or a GMO. A GMO is an organism that has had new genes transferred into it by genetic engineering. Now, we use lots of GMOs in our everyday life, and we use genetic engineering in order to make our lives a bit easier or allow us to gain things that we otherwise wouldn't. So this is frequently used in plants, but it is also used in other conditions. So we can say that we genetically engineer, and I'm putting this in regs, it's important, we genetically engineer many organisms to make GMOs. So we make lots of genetically modified organisms. Some examples are shown on the PowerPoint that I've shared with you. The first one we're going to talk about is corn. Okay, corn is the plant that's grown the most in the US. It's the backbone of the farming industry in the US. In the United States, they use corn to make something called corn syrup, which is added to pretty much every single food substance that the US sells, or any sort of, what's the word I'm looking for? Not produced, sort of man-made food substances. My mind's gone blank. Okay, so it's added to lots of foods. So we can say that corn is genetically engineered in the USA for insect resistance. For insect resistance. Okay, so farmers in the US use corn that has a gene in it that repels insects. If they didn't do that, there would be lots of insects eating their corn, and therefore they won't grow enough. So we can put underneath here, without this gene for insect resistance, the corn would be eaten. So if they didn't have this insect resistance, lots of insects would eat the corn before the farmers ever get to sell it. Another example of organisms that are genetically modified are plums. Plums are a fruit, and we genetically engineer plums so that they are resistant to viruses such as tobacco mosaic virus. Okay, so plums are genetically modified plums are genetically engineered to be resistant to certain viruses. So we can give plums a particular gene which makes them resistant to viruses, and therefore they won't die of the virus. So we can say this stops plums dying of the virus. And that way we can grow lots of plums and we can sell them and eat them. Another example that I researched was soybeans. Soybeans make up a large part of the, particularly a vegetarian's diet. They're added to lots of things and they make up soy. They make up soy protein. Okay. Now these have a herbicide tolerance. So soybeans are genetically engineered to be resistant to herbicides. Now, herbicides are chemicals that we spray plants with to kill weeds. So if you're a farmer, you want to spray herbicide across all of your land. You only want to kill the weeds. You don't want to kill the plants you're trying to grow, such as the soybean. Therefore, if your soybeans are resistant to the herbicide, you haven't got to worry about them dying. So herbicides are chemicals used to kill weeds. So we use herbicides to kill weeds. However, herbicides also kill good plants that we want. So if we make the soybeans resistant to herbicides, we can spray them with it without worry of them dying. Final one, and I'm sure this is the one you've all been most interested in. I've put a picture of a cat here, and we're not going to write this down, but I just want you to listen. When I was at university, there was not at my university, I can't remember which university it was, but there was a scientific experiment where scientists were looking to find a cure for HIV, human immunovirus. And what they noticed was that jellyfish were actually immune to HIV. For whatever reason, the HIV virus couldn't infect jellyfish. So what scientists did was they removed the gene for this HIV resistance from the jellyfish and they put it into a cat. They genetically engineered the cat to see if the cat would then become resistant to the cat version of HIV, which is known as FIV, feline immunovirus. Now, look, interestingly enough, the experiment worked. The cats were no longer able to be infected by FIV. And everything seemed great until the evening when the scientists went to leave. When they switched off the lights in the room where all the cats were kept in cages that had been genetically engineered, they noticed that all of the cats glowed in the dark. Now, this is because not only did the gene for HIV resistance stop the cats getting HIV or FIV, but it also caused their bodies to produce a protein that made them glow in the dark. And it's important to understand that genetic engineering and forming genetically modified organisms can sometimes have side effects that you're not intending to happen. For example, the cat became a GMO. The cat was a genetically modified organism because we'd used genetic engineering to take a gene from a jellyfish and put it in the cat. And we expected that it would get resistance to HIV, but we didn't expect that it would glow in the dark. Now, if this happened, what else is happening with all of these ones that we happily eat? So you guys might eat corn, you might eat plums, and you might eat soy. How do you know that the genetically modified organisms from which these fruit and vegetable came from, and legumes, how do you know that they haven't had any other side effects like the cat glowing in the dark? And these could have a negative impact on your body. This is why people have a lot of problems with genetic engineering. So, another example of genetic engineering that we can talk about that we haven't mentioned yet, and one that's a bit closer to home potentially, is that bacteria, E. coli, can be genetically engineered to produce the hormone insulin. So I'm going to put in red because it's important. I'm going to put E. coli. The E is a capital, the C is a lowercase, and because it's a bacteria, we underline it. Whenever you write a bacteria name, you underline it. However, if you're typing it on a computer, you put it in italics. So E. coli bacteria can be genetically modified to produce the hormone insulin. Now, in the early 1900s, when scientists first discovered or started understanding what caused diabetes, they knew there was a need to get lots of the hormone insulin so that they can treat people who happen to have diabetes. And what they used to do was they used to take the insulin from dead bodies, from cadavers. They used to take it out of the pancreas from cadavers. They also used to take insulin from pig cadavers, and they would use insulin from a pig in order to help people with diabetes. However, as the years have gone on, more and more people have become diabetic, and therefore there's been a greater need for insulin. Lots of people need insulin in order to survive. And now we use genetic engineering to genetically modify E. coli to produce insulin. And we're going to talk now about how that takes place. Okay? So we're going to keep, if you need about half a page in your book for this, okay? So if you haven't, I suggest you go on to the next page. We first of all need a copy of the human insulin gene, okay? So this is the insulin gene in red. Insulin gene. And this is found with human DNA as normal. In your DNA, there would be a gene for insulin among other genes. So in blue, we've got the human DNA. Okay? Now, what we can do is we can use something called restriction enzymes to remove this insulin gene. Okay? So I'm going to put on the arrow because restriction, I'm putting it in green because it's a keyword. Restriction, R-E-S-T-R-I-C-T-I-O-N, restriction enzyme. So we can put here number one, a restriction enzyme is used to remove the insulin gene from human DNA. Okay? So a restriction enzyme is used to remove the insulin gene from human DNA. A restriction enzyme is an enzyme that cuts DNA at specific base sequences. So an enzyme that cuts DNA at specific base sequences. So, for example, you can get a restriction enzyme that will cut the DNA wherever there is an ACCC. Okay? So if we know that at one end of the insulin gene there is an ACCC, at the other end of the gene there is an ACCC, we can use that restriction enzyme to cut the DNA so that the insulin gene can be removed. Okay? There are millions of different restriction enzymes which allow us to cut DNA at specific points. Okay? What we then do is we get a bacteria. So, in fact, I'll use a different colour. We've got a bacterial cell. Inside that bacterial cell, there is what's known as a plasmid. In fact, I'll use a different. What colour can I use? I want one that'll stick out. We'll do that colour. Inside that DNA, in that bacterial cell, there is a plasmid. We can remove the plasmid from the bacteria. And then we can cut open the plasmid using the same restriction enzyme as we used here. And what will happen is you'll be left with a plasmid that looks like this. So this is a bacteria or E. coli. Okay, this is my plasmid. And here we're going to use the restriction enzyme again. So on this arrow here, I'm going to put restriction enzyme. So number 2, we can say a plasmid. And if you can't remember what a plasmid is, you need to go back and look at your notes on cells. A plasmid is removed from an E. coli bacteria and cut open using the same restriction enzyme, used to remove the insulin gene. From human DNA. So we've said that a plasmid is removed from an E. coli bacteria. It is then cut open using the same restriction enzyme we used here so that it is now open. Again, if you don't know what a plasmid is, you need to look back at your notes on cell structure, but a plasmid is found inside bacteria and it is a single loop of DNA or genetic material. And this genetic material can be swapped with other bacteria to give them new characteristics. Now, what's done next is that this open plasmid has the human insulin gene added to it with use of an enzyme known as DNA ligase. So this is added to this, like so. So we can say number 3. And we can also put on the arrows, in fact, before we do that, DNA ligase. So where I'm going to put it, DNA ligase, Ligase. So number 4, the plasmid has the insulin gene added to it using DNA ligase. DNA ligase. Now, DNA ligase is an enzyme that sticks genetic material together. The word lig means to stick. So DNA ligase is defined as an enzyme that sticks genetic material or DNA together. Okay, so it's an enzyme that sticks DNA together. What then happens is that we put this plasmid into an E. coli bacteria, like so. This bacteria can now produce insulin because it has the gene for insulin inside of it. So we can put over here E. coli with modified plasmid can now make insulin. Okay, so E. coli with modified plasmid can now make insulin. So for number 4, sorry, number 5 even, that should be number 3, apologies. This is number 4. So number 4, the plasmid, or the modified plasmid, is inserted into a E. coli bacteria which now makes insulin. Okay, so this bacteria now makes insulin. Now the bacteria will then divide. It will go through binary fission. Again, something If you don't know what it means, you need to look back at your notes for cell structure, particularly bacterial cells. So binary fission. And it'll form lots of E. coli bacteria, which all have, it's the worst oblong ever, which all have this modified plasmid in it. And they can all make insulin. Okay? So the bacteria will divide to form lots of bacteria, all with the insulin gene inside of it, and therefore they can all make insulin. Okay? So number 5, I'll just write it down here. Number 5, the bacteria or E. coli reproduces by binary fission. Right then, just enough room. So, E. coli can be genetically modified to produce the hormone insulin. What we do is we remove the insulin gene from human DNA using a restriction enzyme. A restriction enzyme is an enzyme that cuts DNA at specific sequences. We then remove a plasmid from an E. coli. We cut it with the same restriction enzyme to open it up. We then add the human insulin gene to the plasmid via DNA ligase. DNA ligase is an enzyme that sticks DNA together. We then put the plasmid inside of an E. coli bacteria. This forms a E. coli which is able to produce insulin. This E. coli then goes through binary fission to produce lots of E. coli that all have the insulin gene in them and all produce insulin which we can then scoop up and we can use. Now, there is lots of research into how genetic engineering can be used to cure illnesses such as cystic fibrosis. So in the last lesson on genetic crosses, we spoke about cystic fibrosis and how it's a pretty horrible illness. And we can use genetic engineering, hopefully, to put a healthy gene for where cystic fibrosis is into sufferers in order to stop them having the illness. However, this is highly experimental at the moment. It's not nothing that's used widely. However, it is a hope in the future that genetic illnesses such as cystic fibrosis and Huntington's disease can undergo this process where humans can have their DNA altered so that they no longer have that genetic illness. So you should now be able to tell me what a genetically modified organism is. You should be able to explain the process of genetic engineering to produce genetically modified organisms, and you should be able to evaluate how genetic engineering could potentially be used to cure people with inherited disorders. Should you have any questions about today's lesson, please do not hesitate to contact me via the usual means, and if not, please take care. I will see you in the next recording.